Rheumatoid arthritis is a lifelong autoimmune disease characterized by the presence of synovitis, cartilage damage, bone damage, followed by deformation of the joints. These are some of my disclosures. Alternative pathway plays a crucial role in the pathogenesis of the collagen antibody-induced arthritis, a mouse model of human rheumatoid arthritis. Factor H plays an important regulatory role in CAIA. However, our knowledge of the presence and roles of Factor H and Factor H related genes and proteins in the synovium of patients during early and chronic RA is lacking. We have previously explored the levels and distribution of complement activation, regulatory and effector mRNAs and proteins in the synovium of patients with RA and demonstrated relationships between increasing disease activity and alternative pathway gene expression. The synovium of patients with RA has been classified into three different cellular pathotypes, namely lymphoid, myeloid, and fibroid, but it is unknown what might be the drivers of these three pathotypes. There is uncertainty regarding the in vivo role of factor H related proteins compared to factor H protein in the pathogenesis of non-renal, non-eye tissue diseases such as RA. There were three main goals of this study. The first goal was to determine, using multiplex immunohistochemistry and specific antibodies in which cells and location in the synovium, different factor H related and factor H proteins are expressed and located in RA patient. Second, to explore using bulk mRNA sequencing the level of expression of the 5 human factor H related mRNAs present in the RA synovium and in blood as related to different pathotypes and clinical disease severity. The third, using the global factor H knockout mice created in Colorado, explored the role of factor H related genes and protein in the development of CAIA and by inference RA. In order to find out the answer for all of these questions, we obtained synovial biopsies by using an ultrasound guided technology. Synovial biopsies from early RA patients were used from PEACE clinical trials, and synovial biopsies from chronic RA patients were obtained from STRAP clinical trial. And some of the biopsies were also obtained from Accelerating Medicine's partnership clinical trial just to standardize the various antibodies factor H related protein. 6 to 8 synovial biopsies were obtained from each patient, and the size of each biopsies are from 0.3 mm to 5 mm. Then half of the biopsies were fixed in the formalin for histopathology and multiplex immunohistochemistry, and the other half of the biopsies were fixed in the RNA later buffer for bulk RNA sequencing analysis using 10x genomic technology. We found out that factor H related 2 and factor H related 3 proteins were abundantly present in the synovial biopsies of RA patients as compared to factor H related 1, 4, and 5. Factor H related 5 was always minimally present. On the other hand, factor H as well as C3C were abundantly present in the synovium. One of the most important observations we have made that in the synovial lining particularly, factor H was totally absent. In contrast, it was abundantly present in the subsynovial lining area, indicating that the alternative pathway of the complement system can get activated at very early stage of the disease, and it's the synovial lining where most of the rheumatologists believe that the disease initiates. Then we ask the question which specific cells express high and low levels of factor H related proteins. Here we found out specifically the macrophages, they highly express factor H related 2 and factor H related 3 proteins as compared to 1, 4, and 5. As I mentioned, factor H related protein 5 was minimally present. On the other hand, factor H related 2 protein was abundantly present in the fibroblast. On this side, I'm showing a multiplex immunohistochemistry image and showing the presence of all factor accelerated proteins by using a false color coding scheme. For example, factor accelerated 1 protein has been shown in a magenta color, and factor accelerated 2 has been shown in a white color, and factor accelerated protein has been shown which is abundantly present in the subsynovial lining area in a cyan color, and factor accelerated 3 protein has been shown in the subsynovial lining area in a green color. Another observation we have made that factor accelerated proteins were expressed by the same cells expressing factor H protein. For example, here in the subsynovial lining area, factor H related 2 proteins were expressed, not only factor H related 2 but also factor H, and sometimes we have seen that same cells were expressing factor H related 1, 2 and factor H related 4 protein. Another observation we have made that factor H related 4 protein were abundantly present in the synovial lining areas. And these data clearly shows that there is a regional co-localization of factor H related proteins in the subsynovial lining area along with factor H. Then we ask the question which specific pathotype express high or low levels of complement factor H related genes, and here we found out that there was no statistically significant difference in all of these pathotypes in the synovium as well as in the blood. The top box plots above this red line showing the gene expression of factor H related genes in the synovium, and the bottom one shows in the blood. And we have seen that there was no detection of factor H related 5 gene in the blood consistently in all the samples. Then we also looked at the correlations or the association between -28 C-reactive protein and various factor accelerated genes, and in the synovium as well as in the blood of early RA patients, since these were paired biopsies as well as the blood samples from the same patient. -28 C-reactive protein is one of the ACR approved criteria to measure the clinical disease score activity in early as well as chronic RA patient. Here we found out there was no correlation or the association between -28 C-reactive protein and various complement protein gene expression in the synovium and also in the blood, except factor accelerated 3 gene, which was not only positively correlated but also it was statistically significant. Here I'm not showing the data regarding the factor accelerated 2 gene expression because it was not present in either of all of the database from the 3 clinical trials. Again, we could not detect the bulk RNA sequencing in the PBMCs form in all of these patients in early RA, but it was present in the blood in the synovium at very low level. Then we looked at, based on these studies, we looked at the correlation between rheumatoid factor and factor accelerated 3 gene expression and anti-CCP level and factor accelerated 3 gene expression. Rheumatoid factor and anti-CCP levels are being used as standard biomarker to diagnose this disease in the early as well as in the chronic RA. Here we found out the correlation between rheumatoid factor as well as anti-CCP and complement factor accelerated 3 gene expression was not only positive but also highly statistically significant in all 208 patients. So these data clearly shows that complement factor accelerated 3 gene of the protein might be a pathogenic protein or become pathogenic from early to chronic RA during the evolution of the disease. We also looked at the correlation between axillary erosion, which shows the overall condition of the joint, and complement factor accelerated 3 gene expression, and also the correlation between axillary joint space narrowing, which shows the deterioration of the cartilage, and complement factor accelerated 3 gene expression. Here the correlations were again not only positive but also highly statistically significant, confirming that factor accelerated 3 gene or the protein is in fact a pathogenic protein. Interestingly, we found out there was a negative correlation between DASH28, C-reactive protein, and factor H gene expression in the synovium, and there was no correlation in the blood. These data clearly show that although factor H gene or the protein is increasing with the disease, but it is not enough to keep up with the disease, and this is in contrast to factor H later gene expression in the synovium. In order to provide the proof of principle that factor isolated genes they plays an important role in the pathogenesis of arthritis, we use factor isolated knockout mice, and these has been created by Dr. Hollers in collaboration with Dr. Josh Thurman in Colorado. Here, the entire tiny locus of factor isolated genes have been removed by using CRISPR-Cas9 technology, and these are known as global factor H knockout mice. One of the most important characteristic of these mice is that these mice have identical levels of C3 in the circulation as compared to factor H, which are slightly higher in the circulation, and these were statistically significant. Then these mice were subjected to collagen antibody-induced arthritis, and mice were injected with a mixture of 5 monoclonal antibodies known as arthritomab, followed by injection of LPS at day 3. All mice developed disease. Interestingly, there was more than 80% reduction in the clinical disease score activity in global factor H knockout mice as compared to the wild type mice. Consistently, there was also a decrease in histopathological scores for inflammation, pannus formation, cartilage damage, and bone damage in global factor H knockout mice with disease compared with the wild type mice with disease. And also we found out there was a less 80% less C3 deposition in the synovium as well as on the surface of the cartilage in global factor H knockout mice with disease as compared to the wild type mice with disease, indicating that factor H related genes and the proteins they modulate the alternate path for the complement system. And also there was more than 60% decrease in the number of macrophages in the synovium as in the global factor H knockout mice as compared to the wild type mice with disease. Finally, the dispersed but differentially cell associated presence of factor H related proteins in the synovium of early RA patient suggests that these proteins are involved in the pathogenesis of RA. Factor H, in contrast to factor H related proteins, is almost absent in the synovial lining but more abundantly present in the subsynovial lining area, a phenotype that could lead to the dysregulation of the alternate pathway in the lining at very early stage of the disease. Factor H related 3 mRNA levels are positively correlated with rheumatoid factors, anti-CCP and X-ray erosions, and thus could be a new biomarker and important pathogenic protein of RA. In contrast, factor H mRNA levels are inversely associated with the disease. Mice lacking global factor H-related expression are strongly protected from CAIA, suggesting that factor H alone could inherently locally control a classical lectin alternate pathway driven tissue disease in the absence of factor H-related protein effects, and emphasizing the key role of complement pathway amplification in the tissue engineering. Finally, I would like to send my sincere thanks to Dr. Michael Hollers, who have provided tremendous support for these studies, and also to other faculty members who have provided synovial biopsies for all of these from all of these clinical trials, and to Dr. Josh Thurman, who have generated global factor H knockout mice in collaboration with Dr. Holler as a part of separate project, and to Dr. Myles Lewis and Pitzalis and Rachel Law from UK, who have provided synovial biopsies from 3 different clinical trials and also share the full access to the clinical data or the information, and to Dr. Paul Morgan, who have provided us complement factor H related 4 antibody which works great in the synovium. Of course, to the NIH for the funding of this project for the last more than 25 years. Thank you very much. If you have any question, I will be happy to answer.